Thank you so much for inviting me to speak today. My name is Kathy Hernandez, and I'm a graduate student, just finished my 5th year in the Koskela lab at UC Berkeley. And I'm going to be talking to you today about my dissertation work exploring bacteria and phage interactions and coevolution in the phyllosphere, which is the above ground surfaces of plants. I lost my cursor. All right. And so I'm generally interested in how environmental context can impact bacteria and phage interactions. And what I love about working with these systems is that you can grow them really simply in the lab in a culture like this, or you could add some spatial structure, grow them on a petri dish and have them interact there. You can also manipulate whether there's spatial structure in the test tube or not by shaking it. You can also manipulate the quantity and quality of resources that are available to the bacteria that you're growing. Or you could also play around with adding different microbes and seeing how that impacts pairwise coevolution, or maybe a network of interactions between bacteria and phage. And so all of this work has been done in vitro and has been shown to be important for shaping bacteria-phage interactions and coevolutionary dynamics. But what's less clear is how our understanding of the way that spatial structure, resource availability, etc., plays out in vitro might translate to in vivo host-associated environments. And in particular, bacteria-phage interactions on plants is highly understudied. And so I specifically work on Pseudomonas syringae, which is a bacterial plant pathogen that it's described as a species complex that among the various strains infects a wide variety of different agriculturally important plant hosts. And we work specifically on tomato in the lab. And so for its infection cycle, it begins as an epiphyte growing on the leaf surface, where it multiplies, and then it can induce stomatal opening, or it can enter via wounds. And then it enters into this light green space here called the apoplast, which is an intercellular space that's typically air-filled. And then it multiplies pretty aggressively and causes eventually death of the tissue, so in these brown areas with a chlorotic yellow halo surrounding that. And then this is a picture of what that looks like on tomato leaves. And in terms of phages in the phyllosphere, we don't know a ton, but we do know that they can be isolated from both healthy and diseased leaf tissue. And some former work from a graduate student in our lab was shown to impact bacterial abundance and composition in the microbiome. And some former work from my advisor had shown that they can also be locally adapted to bacteria from the same plant. So we think they're playing a role in the microbiome in the phyllosphere. And so my overall question of interest in my dissertation work has been, how does the phyllosphere impact Pseudomonas syringae phage interactions more in a pairwise way than throughout the whole microbiome? And so I'm going to tell you about 4 different projects briefly that are all tied together that fall under this umbrella of question. And so the first is a bacteria-phage coevolution in vitro project, just to give us a baseline understanding of how this organism might co-evolve with phages in the lab. And then the next is a direct experimental coevolution comparison of how coevolution plays out both in vitro and in plants. And then the next are some newer unfinished work exploring some follow-up questions from the results from that second project. And the first is whether there might be plant cell phage direct interactions, which is very exploratory. And then the last one is on biofilm phage interactions and whether the Pseudomonas syringae biofilm might be impacting rates of phage replication in this system. And so for this first project, our question of interest is fairly simple. What are the coevolutionary dynamics like in vitro for Pseudomonas syringae and a single phage that we typically work with in the lab? And so as a primer on the major types of host parasite coevolutionary dynamics that people talk about, so if we imagine starting with this sensitive purple bacteria that can be infected by this purple phage, it'll then be selected to evolve resistance, and then say it has this pink mutation that now allows it to be resistant to that purple phage. If the phage co-evolves, it could potentially expand its host range and now become this pink-purple phage that can infect this new host and infect that old purple host. Alternatively, you could get this novel pink type that instead the phage evolves to infect this pink type but cannot infect that old purple type. So there's no change in host range there. And so if you follow out the dynamics on the top, what you get over coevolutionary time is a really resistant bacteria that's expanded its resistance range quite a lot, and a phage that's expanded its infectivity range, and we call this arms race dynamics. And on the bottom, if you follow those dynamics out, if you stop it at some point, you're going to get just some dominant bacterial type that's different from the other bacteria that you might have seen throughout coevolution and a different phage type. And so at the end or whenever you stop your coevolution experiment, you have different predictions about what the ranges of these phages might look like and the host resistance ranges. And these bottom dynamics we call fluctuating selection dynamics. And you can think of these as sort of extremes of a continuum. And so what do we see when we actually do bacteriophage coevolution in the lab? We often see arms race dynamics, but sometimes the bacteria win. So they evolve resistance in a way that the phage can't counter-adapt and be able to infect, and then they just decay. But this also depends on environmental conditions, like I said earlier. So if you alter the resource level or whether you add spatial structure, this can also impact the dynamics. And it also depends on length of coevolutionary time. So a lot of these host parasite coevolution experiments with bacteria and phage are relatively short still, but this paper was really cool because they looked at how the coevolutionary dynamics changed over time in a longer-term bacteriophage coevolution experiment. And so this is time on the x-axis, and on the top panel we have infectivity of the phages, bottom is resistance of the bacteria, and what they found was that initially there's an increase in the range of infectivity and resistance, so it looks like an arms race, but eventually it plateaus and looks more like fluctuating selection dynamics, and that was due to costs of increasing range beyond that point. And so costs are really important, and they can prevent an arms race from becoming this sort of infinitely escalating arms race. And so we were interested in exploring these sorts of dynamics and questions with our Pseudomonas, syringae, and phage system. And so we co-evolved, co-passaged our bacteria and phage together just for 6 transfers, so that's a relatively short coevolution experiment. And then we wanted to look at host range and whether that might be coming at a cost in our co-evolved phages. And so on the x-axis here, we have host range of our phages, and on the y-axis is a sort of measure of cost, which is how well they can kill bacteria in a growth curve. And then I'm going to point you just to a couple of the treatments here. So the ancestral phage is in pink, and the co-evolved phages are in green, and we'll ignore the evolved phages for now. And so the ancestor is over here, so it can infect 20 of the colonies we tested. All the co-evolved phages can infect more. But we don't see an evidence of a negative relationship between host range and the ability to reduce bacterial growth. And so it doesn't seem like we're seeing a trade-off, but the phages have expanded their host range. So it kind of looks like an arms race, maybe without cost. But like I said, this is a short-term project, so maybe something would be different if we expanded those dynamics out for longer. And then on the flip side of this, in terms of bacterial resistance and whether that might be coming at a cost in this system. So this is some work from a former graduate student in the lab, now a postdoc, Sean Meaden, who was looking at the cost of being resistant to phage and whether that was context dependent. And so what he did was he selected for resistant bacteria in the lab, and then he grew them in the presence and absence of phage in vitro and compared that to the growth of sensitive bacteria. And so on the left here, this is in vitro with phage. The resistant ones are growing better than the sensitive ones, as you would expect, they're resistant. And then here we have in vitro in the absence of phage, and there was no difference between the resistant and sensitive bacteria here, and so that seemed like there wasn't a cost in vitro for this type of resistance. However, when he grew these bacteria in plants, what he found was that the sensitive bacteria are that top line, and the bottom is the resistant bacteria. And so here he did find that the resistant bacteria seemed to be growing more slowly and reaching a lower final density at his last sampling point. And so this told us that there is this context-dependent cost of being a resistant bacterial cell in planta compared to in vitro. And so what we then wanted to ask as a follow-up to this was, how does the plant environment impact de novo evolution of resistance to phage? So in that prior work, he was selecting for resistance in vitro and then growing the bacteria in both environments. And so we wanted to say, okay, if we start with sensitive bacteria and phages that can infect them, put them in both environments, how do the coevolutionary dynamics play out given that there's a context-dependent cost? And so we did an experimental coevolution project in both environments where we inoculated plants and liquid cultures with the same starting colony of bacteria and plaque phage. And then we would incubate them for 72 hours, allow them to grow up, and then we would sample for bacteria and phage, and then you separate them out, and then we can recombine them in different combinations. So we can either passage the bacteria or the phage by itself, or co-evolve the two of them. And then we would regenerate inocula, inoculate them again, and we did this for 6 transfers, like in the previous experiment. We had a bunch of different treatment combinations, but the most important ones are B only, which is bacteria that was just passaged by itself without phage. And then we co-evolved bacteria and phage, so co-passaged them together. And then we had 2 treatments where we either just passaged the phage or the bacteria. And then what we would do is we would add the ancestral other partner back in. And what this kind of design allows you to do is really get a sense of what's unique about coevolution in your system compared to evolving against a phage just in general. All right, and then our first question of interest at the end of this was, did they evolve resistance? And so for this, we did a simple streaking assay where you take a liquid stock of phage and you drip it down an agar plate, and then you pick individual colonies of bacteria and you streak them across the line of phage, and if they can grow over the phage streak, they're resistant, and if they can't, then they're sensitive. And so this is an example of what that looks like on an actual agar plate. And then in this graph, we have the proportion resistant for both the in vitro passage populations and the in planta passage populations. We have our different treatments that I had told you about on the previous slide on the x-axis. And then we assayed for resistance against 2 types of phage, so the ancestral original phage from the experiment and contemporary phage. So that's phage from the end of the experiment. And so for the in vitro populations, the 3 that I've marked with phage here were all passaged with phage in some form. And this B-only population was just bacteria, so they weren't resistant to the phages at all. And then these were all resistant to some degree. But in contrast, what was really surprising to us was that we didn't find much resistance at all in our plant passage populations in any of the populations that were passaged with phage. And so this was really surprising to us. We did repeat this experiment a few times because we were kind of shocked. And we also did an in planta resistance assay that I'm not really going to talk about because we thought that maybe this in vitro streaking assay was too artificial. We weren't capturing the resistance that might be manifesting somehow in the plant environment, but they didn't seem to be resistant there either. And so our next question was, what do the phage replication rates look like in this environment? Because it was unknown. And so we took some bacteria from the end of the experiment that had been passaged in planta, a couple different types, and we grew them in plants with phage and looked at how well the phages replicated and compared that to phage inoculated in plants by itself to see if we were getting any phage decay in this environment. And so these are the phage densities over time. Time here is on the x-axis, and then I'm plotting phage density. And then the 3 different point types. So the white points are phage only, so that was like our phage decay control. And then we have B-only bacteria, so that was the bacteria that should be sensitive, was never passaged with phage. And so phages should be able to replicate on that bacteria. And then B-evolved bacteria, which we think is not resistant, but if any treatment were resistant, it would be that one because it was passaged with the ancestral phage repeatedly. And so here's what we found. So the white points here, there was no change over time in the phage signal in the plants, so it didn't seem like there was much phage decay. And then in the 2 treatments with bacteria where the phages could have replicated, we did see a significant increase in 24 hours and then no change between 24 and 72 hours. But this is a really low rate, so it's not even an order of magnitude. So compared to what's happening in vitro, this is really low phage replication rates. And it didn't seem like there was a difference between these 2 treatments, which again suggests that they can replicate just as well on both types of populations. And so our final experiment as part of this project was trying to get a sense of whether there's a benefit to being a resistant cell at all in these environments, especially when you're under phage selection. And so we selected for resistant and sensitive colonies in vitro for this assay. And then we grew them, the resistant and sensitive bacteria, in the presence and absence of phage in both environments, so lots of different combinations. And so here on the left panel, we have in vitro growth of these bacteria. On the right, we have in planta growth of these bacteria, and these are the same colonies, same resistant and sensitive colonies that were grown in both environments. And then time on the x-axis, and then absence of phage is in black and presence of phage is in gray. And so yeah, resistant and sensitive in each panel. And so in vitro, we found with the resistant colonies that they grow equally well in the presence and absence of phage, makes sense. Sensitive colonies, they grow, doesn't seem to be much of a cost of resistance if you compare the absence of phage growth between resistant and sensitive colonies here, but we do see that the sensitive ones are growing, reaching lower densities in the presence of phage, and that's what you would expect. And then in planta, it was similar, but So with the resistant colonies, they do appear to be resistant. They're growing pretty similarly in the presence and absence of phage. And in the sensitive colonies, they also seem to be growing. They are significantly reduced by the presence of phage at 24 and 72 hours. But the interesting comparison here is if you were trying to imagine a sort of growth rate for these sorts of resistant and sensitive colonies, the resistant ones grow much faster in vitro than the sensitive ones in the presence of phage. But if you make that same comparison in planta, they're growing pretty similarly. And so that suggested to us that these in vitro selected resistant mutants have a fitness benefit over sensitive colonies when they're growing in the presence of phage in vitro, but not in planta. And so some conclusions from this. The rapid evolution of resistance in vitro doesn't seem to be a very reliable model for what happens in planta. And it's not to say that resistance is ever going to evolve and maybe would spread a little later, but it's certainly not at the rates that we're seeing in vitro. Phage replication in the apoplast is occurring at a very low rate, and resistance to phage seems to be beneficial in vitro, but less so in planta. And this work was published last year if you're interested. All right, and so a couple follow-ups from that. I am really interested in this question of why we're not seeing so much phage replication in this environment. And so one idea is to see if there's any interesting direct interactions between plant cells and bacteriophages. And so the questions here were whether plant cells might be directly responding to or uptake phages, maybe make them unavailable to infecting bacteria in some way. And so this work was really inspired by work that was done in animal cell systems, both associated with Jeremy Barr, who's at Monash University in Australia. And so this left project was looking at the crossing of bacteriophages through animal cell layers. And so they can preferentially move in a certain direction. And you can also find them localizing within these animal cells. So this is the outline of the cell in red, DNA in blue, and these are phages stained in green. And then on the right over here was another paper where they were looking at how phages could stick to mucus that's produced by epithelial cells, and it can protect the underlying cells from pathogenic infection. And so I really wanted to see if we could try to do something like this in plants. And so instead of doing this with phyllosphere cells and having to do tissue culture, I instead wanted to work on root border cells, which are a specialized type of root cell. And I did this work in collaboration with Jeremy and one of his graduate students, Marion, at Monash. And so this is a root tip. And so these are root border cells that are being kind of sloughed off when the root tip comes into contact with water. And they're involved in protecting the growing root tip from pathogens and contaminants in the soil. And what they do is they can form this, what's called an extracellular trap that can trap contaminants, and they also respond directly to the presence of bacteria in the soil and can expand this trap. And we also know that in the soil, there are a lot of phages, so they can outnumber bacteria 100 to 1. And so I wanted to know if we could try testing some of those same questions. Do phages maybe impact this extracellular trap in any way? Do the cells respond? And can we find phages inside of root border cells potentially? And so I worked with corn for this because they make nice large root tips and nice root border cells. And I incubated them with various compounds. And then I looked at a proxy for the amount of extracellular DNA, which is a really important component in these extracellular traps. And looked at how that the amount of extracellular DNA changed over time with these various compounds. And so this is a water control, didn't really change. This is DNase, it degraded the extracellular trap, that's what it's expected to do. And then here we have two bacterial treatments, so E. coli and heat-killed E. coli. And so that did stimulate an increase in the extracellular trap, and that's what we would expect as well. So that told me the assay seemed to be working. And then here are my phage treatments. So I had T4, which is a model phage, heat-killed T4, and T4 filtrate. And the T4 filtrate is an important control because it's essentially the liquid that the phages are prepared in that has some cellular debris components in it. And so it's not, the phages that we prepared were not entirely clear of like LPS and those other components of bacterial cells that might be stimulating the root border cells themselves. And that did seem to stimulate these cells maybe a little bit and similar to the phages. So it seemed to us that the phages themselves are probably not stimulating any changes in these extracellular traps. And then we also did some incubations of root border cells with phages, similar to the way they did with the animal cells. And I stained the phages here with the green, and then the cell outlines in red, and the blue is DNA. And so it did appear that there might be phages within these root border cells. And so this is unfinished new work that I'm super excited to potentially follow up on and kind of ask what rates these phages could be entering root border cells and whether they're inactivated in that process. Yeah. And then, so for the final question, I wanted to look at how the Pseudomonas syringae biofilm might be impacting phage replication rates in planta. This biofilm formation is pretty important for this pathogen, and we know from in vitro work that that can impact phage interactions. And so when the pathogen first enters into this apoplast space, it starts as a motile planktonic cell that then attaches to the plant cell, and then it produces its biofilm. And like I said, this can limit the phage access to bacterial cells that rely on diffusion. It can also impact whether receptors are available for phage infection. And so we want to look at whether this biofilm formation is impacting these replication rates in the plant environment. And so we've done a bunch of biofilm phage interaction experiments in the lab, and I've done this with an amazing undergraduate from Berkeley, Aspen. And so we have done these experiments mostly looking at the biofilm side and seeing whether phage impacts how much biofilm there is. And so we grow the bacteria, and then we put them in these plastic dishes that allow for biofilm formation, let them develop for a period of time, then we add phage, incubate for a period of time, and stain with crystal violet to get a proxy of how much biofilm there is. And so I'll just show you one of our various in vitro trial experiments. And so here we tested a panel of phages and whether they impacted the amount of biofilm in the wells in any way. And so here's our no phage control. That's our strain DC3000 by itself. And then these different colored boxes are all of the different phages that we tried. And so we didn't see any decreases in biofilm production, but we did see some significant increases, which was interesting. And so our final question for this work, we want to see if we can experimentally evolve and select for and against biofilm formation in this pathogen so that we can have populations and colonies that are better or worse biofilm formers that we can then assay in plants. And for this, we're taking advantage of a method developed by Von Cooper and his group where they passage biofilms on beads. So they grow the bacteria in a culture with a bead, and then the biofilm can grow on the bead, and then they can transfer that bead to a new well with a free bead that the old biofilm can then disperse onto. And then you select by serially passaging these beads. And we also selected against biofilm formation by having beads in the well, but transferring the liquid and not the bead portion. And so this is just a picture of those plates. We didn't do it in these tubes because we didn't have the right type of bead and were playing around with methods. And so we did them in these dishes, and we also tested static and shaken cultures because this is pretty new methods development. And so here are two different plate types. We had shaken plates and static plates, and the two different colors are the B are the biofilm selected wells, and the P are the planktonic selected wells. And here I'm showing you the bead CFU over total CFU in the well. And so if they're forming more bacteria or growing more onto the bead, that would seem to suggest that our selection method is working in the biofilm selected populations. And so in the shaken plate, that didn't seem to be true. Didn't seem like our selection method was working very well, but it did seem to work in the static plate. And so moving forward, what I'm hoping to do once I can get back into the lab is take these stronger and weaker biofilm forming populations as well as individual colonies and grow them with phage in plants and quantify phage replication and see whether the amount of biofilm that these colonies and populations can produce has any impact on phage replication. And I'm also interested in quantifying virulence and how diseased the plants get over time, since biofilm formation does seem to be important for virulence in this pathogen. And so going back to our questions of interest, in terms of coevolutionary dynamics in vitro, it looks like we have arms race dynamics, and so our co-evolved phages have expanded their host range, and it doesn't appear to be costly. And then for our second question, how does the plant environment impact de novo evolution of resistance to phage? It looks like there are really low rates of phage replication and potentially a context-dependent cost again, which means that there's not much of a benefit of being resistant. And then do plant cells directly respond to our uptake phages? It doesn't seem like the root border cells at least respond in terms of increased trap formation, but they may be uptaking phages. And then in terms of whether the Pseudomonas syringae biofilm might impact phage replication, it probably does, but that's to be determined with future work. And so, yeah, I'd like to thank the seminar team for inviting me today to speak, and all the members of the Koskela lab, past and present. This is our most recent Zoom photo. And then the Bar lab were super helpful for the root border cell project. And then a bunch of former undergraduates in the lab, Rena, Aspen, Sophie, Corey, and Priyanka, who have helped on various aspects of these different projects. And that's it.
Well, thank you so much, Catherine, for very interesting presentation and study that you showed us. I see a couple of people are having questions. I first go and ask people that raised hand to ask their own questions. We have Adriana Sands. Adriana, if you would like to unmute yourself by clicking on the, your microphone picture, and then you can ask your question.
Yeah, thank you very much. I hope you hear me.
Yeah, it's all good.
Okay. Yeah. So thank you very much, Catherine, for the super great talk. I have two questions. The first question is related to the coevolution dynamics in both environments for phage. And you see that there are no resistant, there is not a cost for resistant work because basically they are not replicating phages. So phages are not replicating in planta, right? So do you know which is the bottleneck there for the populations of bacteria?
Well, the bacteria seem to be replicating. They definitely increase like 3 or 4 orders of magnitude while they're growing in planta. The phages are not comparatively replicating as much as we would expect given the level of bacterial replication. So I don't know. The phages seem to replicate a bit, so they do impact bacterial densities and they are replicating, but just at a very low level. And that replication seems to be happening mostly in the first 24 hours after we've inoculated them. And so that's when the bacteria are most actively growing, so that makes sense. And then we don't see much happening after that. But yeah, the reasons why we don't see it, I mean, the plant environment is so different from the in vitro environment. They're not comparable at all. And it's certainly possible that we could manipulate the in vitro conditions to maybe try and recapitulate some of the important environmental features of the plant. So for example, we could use minimal media that mimics the plant nutrients that are available, which also would lead to biofilm formation, so we can try that. And there's also spatial structure in the plant environment, and there's a lot of heterogeneity. It's really patchy in terms of where the bacteria are growing, all of which I think are all playing a role in why the phages aren't replicating so much. But yeah, I think pinning down the exact reasons why is tricky.
Okay. And sorry, Adriana, are you okay? Is that?
Yeah, I have just another question.
Yeah, sure.
Yeah, it's related to a picture that you show where you treat biofilms with different phages and treatments, and you see that basically when you treat with some strains of phages, the biofilm increased. I don't know if you know the mechanism why is this promoting this increasing of biofilm formation.
No, it's a good question. So I guess I wonder if maybe some cells on the outside of the biofilm might be susceptible to phage, and that could release some nutrients that are now available. I don't know. There's also, so the phage diffusion through biofilms is limited, but can depend on the properties of the phage. And so maybe if the size of the phages are different, there might be different receptors that they're using. I'm not sure, but yeah, the phages are from very different groups, so different phage families. And so, yeah, they could be interacting with the bacteria in very different ways. But yeah, I'm not really sure.
Okay, thank you.
Thank you, Adriana. David Demery has a question I think is related to the same topic. It's about the low rate of phage replication that you mentioned. He's asking, did you try longer experiments and, extend your experimental evolution time, I
Yeah,
believe,
the
or the
experimental,
treatment time?
sorry, yeah, the experimental evolution, no, we didn't do it, longer. But it's certainly possible that maybe eventually we would start to see some increase in resistance. And in terms of like an individual passage, we did 72 hours because that was about the time that the bacteria reached carrying capacity in the leaves, and there wasn't really any more replication, and the tissue was kind of dead at that point. So we think we were capturing what was happening within those individual leaves at least.
That's great. Okay. Steve Diggle, would you like to unmute yourself, Steve, and ask the question?
Hi, great talk, Catherine. Just got a question about your resistant Pseudomonas strains. So have you sequenced them? So do you know what the actual mutations are that allow them to become resistant?
Yeah, it's a good question. So not in my experiments, but in Sean's experiments where he was looking at that context-dependent cost he did, and they were all LPS mutants. So we think that the receptor for the phages is LPS, unlike some other Pseudomonas syringae phage work, where they use Phi6, which I think uses pilus as the receptor. And so, yeah, so we think it's LPS, and I would bet that that's probably what was happening in my experiment as well.
Okay. Do you also follow on to that? Do you find that when you look at the populations of bacteria that you've evolved, you find mixtures between sensitive and resistant strains, so you can have both in the same experiment?
In terms of the in vitro populations or the in planta?
Both.
In vitro, yeah, in vitro we definitely saw, so in that plot I was showing with the proportion resistant, there were some where they were entirely resistant to the ancestral phage, but I don't think in our populations they were all entirely resistant to the contemporary phage. So I do think that like if we were to allow these experiments to play out for longer, that we could get more extended coevolution happening in vitro, which I know is tricky for some bacteria and phage coevolution systems. They sometimes get stuck and the bacteria evolve resistance, like I said, and then the phages can't counteradapt.
Okay. All right. I have a couple of more questions. Joy Leung, if I'm sorry if I'm not pronouncing your name right, and also Madeline May, they both have comments and questions related to increase in biofilm production in your biofilm assay when you added different phages. Joy is asking if is the increase in biofilm production is given some phages strains due to more matrix production or more bacterial cells? And if you want to answer that, and then I ask Madeline's question, which is again related to the same topic.
That's a good question, and I actually don't know. So this is a crystal violet staining assay, and so I guess it could be either, really. But yeah, I haven't quantified the bacterial, like actual bacterial densities in these experiments, and I didn't quantify phage either. So that's stuff that I want to do moving forward to see if we're getting much actual phage replication in these systems, or whether it's just the bacteria responding to phage presence in some way. But yeah, I don't actually know.
And again, related to that topic, Madeline is asking, are there any or at all, or all phages used in your experiment are lytic? And do you think they are inducing SOS response? And does lyting prophages or syringae-syn, syringae-syns, and also she's asking, maybe like she's suggesting that maybe that's why they're increasing the biofilm formation when they're treated with different phages. Do you have any comments? I hope that I asked the question correctly, Madeline.
So all of the phages that we're working with are exclusively lytic phage, and I don't know. In terms of temperate phages, I don't think we've ever seen in the strains that we work with ever any like temperate phages coming out when the bacteria are stressed. So I'm not sure, but I guess it's possible.
Yeah, considering that, like I'm not sure about the prophages in Pseudomonas syringae, if you're working on that. Sorry, I just forgot this correct strain of the bacteria, but in terms of Pseudomonas, we have lots of prophages.
Yeah, no, it's a great question. I think we've kind of, in our lab, mostly focused on like the lytic phages and lytic dynamics. It's easier for these coevolutionary studies and kind of ignored the prophages, but yeah, it's a good question.
The other thing I was wondering, again, related to increase in biofilm, that you mentioned that you're going to check the live cells and the CFUs of the bacteria at the end. It might be due to more efficiency of the phages that are killing the cells, so you get, again, more bacteria cell lysate, so you get more of attachment to the plastic. So I think James Gurney also has a question. Moving on from this section. James, would you like to?
Same set of questions, right? So I got a couple of questions. The first one, that starts from your evolution paper, right? That was the stuff you showed the second stuff. That was the evolution paper you guys did, right?
Yeah.
Yeah. Okay. I was getting horrible deja vu. It was like I've seen this somewhere. Did you guys ever show a way of guaranteeing that you actually had phage bacteria interacting in that sample? Because my understanding is it could just be that the phage didn't see the bacteria.
Yeah, so that's why. No, yeah, of course, that's a great question. Yeah, so that's why we did those phage replication experiments where we took the bacteria. In that case, we took bacteria that had been evolved, but I guess kind of irrelevant. But we did quantify phage replication, and we saw about an order of magnitude increase in the amount of phage that was present over 24 hours, and then no other change. And so it's a really low rate, but we do think the phages are finding some.
Could you not quantify that and say, well, given the birth size and a Poisson distribution, you could work out exactly how many, the MRI, the density of the bacteria, you could work out exactly how many interactions must occur.
Yeah, we do know applied.
You then could pick out how many cells you'd have to pick to actually find a resistant isolate. So I imagine it might be quite low.
We know the applied MOI and we know the starting bacterial density. We actually don't know the sort of phage growth parameters for these phages very well. We've tried to quantify things like adsorption time and burst size, and it hasn't worked very well in the system. I think our phages seem to be really poor adsorbers, even in vitro. And so it's been really tricky trying to get an estimate of, I would really love an estimate of burst size, but we don't have one at the moment.
I think you could fit that with one of the parameters, I guess. But so it kind of goes to my next question of the biofilm stuff. So correct me if I got this wrong. When the syringae lands in the plant, it can either go through one of the pores, and if there's a wound, it gets inside that internal space. That's where it starts making the biofilm, right? It's actually in that internal section where the disease biofilm starts. So how in your system, how are you getting phage from the outside of the plant through that first layer of cell tissue into space?
Yeah, so I didn't talk about the details of the methods for that plant pathogen experiment, but we inoculated directly into the apoplast. So there's a method called syringe inoculation that forces them into that space. And so, we're kind of artificially missing that first layer of the surface interactions where there could be more or less replication. And I do actually think that there could be more phage replication on the surface. I've done experiments with seedlings where I flood them with bacteria and phage rather than inoculating directly into the apoplast, and we see lots more replication there. And so, so I'm interested in following up more on what's unique about the apoplast and why things are so different there. But one big thing is biofilm formation.
So the other thing, and Smil gave you some biofilm suggestions there, saying that potentially it is adaptive response, they're making more polysaccharides or something like that. I mean, that's possible, right? It could happen. The other option is that there is obviously the media has a carrying capacity, and if a phage are selecting the planktonic cells out because they're not in the biofilm, the carrying capacity of the biofilm will be increased. So you'd actually it's an argument from survivorship. You'd see more biofilm because no cells live in the planktonic, so that they all get to use the nutrients inside the biofilm. So you have a larger biofilm at the end. It's not an adaptive response to the phage. It just might be, they can survive, so they get more nutrients at the end.
It's easy to test, I suppose, these. All of this in
easy.
different ways.
Easy. Thank you so much.
Yeah, thank you.
So let's see if there are any questions that I didn't ask. So I think I just have a suggestion, actually, not a suggestion, but the beads that one Cooper's lab use, we use them too, and they're very easy to find. So I can send you the link to where to get them.
Yeah. So I actually had asked him. Yeah, I had asked him about them at a conference once, and he said that like the supplier stopped selling them, and so he had access to them and could send them to people. And I think now the like MIG sequencing center is selling them, so I do want to try using them and see if I get some better results in my experiments.
I think they're just like bigger beads, and they have more surface for bacteria to
and
attach.
They're polystyrene, I'm pretty sure.
Yeah. So it's actually VWR has them, and in the UK we used to just buy them from Beads Shop, which was basically the same and easy to use. But I can send you the link for sure. But we can always.
That's great.
You can always source it from pits as well. Okay, I think this is all for today. Once more, thank you so much for joining us. Feel free to join us in upcoming weeks if you're free. And thank you, everybody who joined us today. Let's once more thank you. And
thanks so much.
Have a great week coming. And people are just telling you, have a great talk. I don't know if you see them in the messages and the chats. That's great. Lovely seeing you, and hopefully you're going to go back to the lab soon and carry on with your experiment.
All right. Thanks so much. Bye.
Thank you. Bye.